Great. Hi, everyone. Thank you for inviting me, and I'm very sorry that I cannot be there in person. Um, I would have loved to be. So today I'm going to talk to you about this delicate balance between weather, diapause, and pest pressures on alfalfa leaf cutting bees. Okay, so as many of you probably know, alfalfa is a huge crop in the United States. Um, it is the third most important field crop economically, both for hay and seed. And the United States in particular produces about 80 million pounds of alfalfa seed each year. If you look at this map that I am showing here on the right, you can see historically the counties in the western US where alfalfa seed was produced. Unfortunately, now, the last several years, alfalfa seed production has really been restricted to these upper northern 5 states. And where I live, right here, if you can see my cursor, in Cache County, Utah, this is basically the only county that's left that's producing alfalfa seed in Utah, in all of Utah. And these more southern US states, Utah, Nevada, California, and Arizona, the alfalfa seed production is all but non-existent anymore. And mainly this is due to loss of acres, but there's a lot of other factors that are influencing why we have a reduction in alfalfa seed. Despite that, it is still a large and economical, valuable crop for most of North America, most of the world. And alfalfa, in particular, as a crop, is pretty unique because it is not reliant, it's not heavily reliant on honeybees for pollination, where a lot of crops are heavily reliant on honeybees, managed honeybees for pollination. And alfalfa is unique because its managed pollinator is Megachile rotundata, the alfalfa leaf cutting bee. And I'm sure a lot of you are familiar with this bee. It's a solitary cavity nesting bee. It's highly important in the pollination of alfalfa seed, and I'm going to go into a little bit why that is. And growers and farmers, at least in the US, are using 100,000 to 150,000 bees per hectare to pollinate their alfalfa. So why use alfalfa leaf cutting bees over honeybees? And honeybees are really considered nectar robbers of alfalfa. Alfalfa is a legume and it creates these pea-shaped flowers. And over here you can kind of see the flowers of alfalfa, and this is a untripped flower, and this is a tripped flower. And what I mean by that is that the heel of the alfalfa flower has actually popped down so that the pollen is now exposed. And honeybees are not don't have the proper mechanics to actually trip that flower, and so they tend to just nectar rob and rather than pollinate. Where the alfalfa leaf cutting bee actually has the right dynamics, weight, and movement to trip those alfalfa flowers, exposing the pollen, and then they transfer and move pollen from one plant to the other, obviously pollinating and creating alfalfa seed. And there's been a number of studies that have shown that alfalfa leaf cutting bees are way more efficient than honeybees are. And this is why the industry as a whole has moved away, and this has been like for decades now that we've moved away from honeybees and are pollinating solely with alfalfa leaf cutting bees. And the alfalfa leaf cutting bee, Megachile rotundata, is considered the most managed solitary bee. Okay, but the US has a big problem because we cannot propagate leafcutter bees. So 90%, probably more than 90% of our bees in the US are imported from Canada. So thank you, you're helping our industry. And so we get we have lots and lots of issues of propagating and maintaining healthy populations of alfalfa leaf-cutting bees, which means that every year we import more and more leaf-cutter bees from Canada. Okay, so why are there challenges with propagating leafcutter bees in the US? And I'm gonna kind of briefly go through these issues, and then I'll talk about some very specific ones that I have investigated. So this idea that these bees can have facultative diapause, so they can either diapause or not diapause. You probably may have also heard this as second generation. There are loads and loads and loads of parasites in the US that attack leaf-cutting bees. We have pollen ball problems, where the egg or early instar larva never develops. Shockroot problems, which is a fungus that infects the larva stage of leaf-cutting bees, and obviously there are always pathogens that have an effect. For my talk today, I am going to primarily focus on these first two things, this diapause and second generation, and parasites. And so I'm gonna give you two research projects that I have been working on recently, and then I'll kind of wrap everything up at the end. Okay, but first, if you're not familiar with the life cycle of an alfalfa leaf cutting bee, we're just going to go through this really quickly. Alfalfa leaf cutting bees overwinter as prepupa, so this is just the last stage of the larva inside a cocoon, and then throughout the winter they stay as the prepupa, and then in spring, once temperatures start warming up, they're going to turn into the pupal stage, and then finally in midsummer they're going to emerge as adults and forage and lay eggs. And so this mom is going to go and lay eggs, and at this point the offspring of this mom have two options that they can do. They can either diapause, so that means that the mom lays the egg develops into a larva, eats all that provision that the mom made, and overwinters as this prepupa, like where we started from the beginning. And then the next year becomes a foraging adult. Alternatively, they can have this strategy where they choose to not diapause, and the larva develops, becomes a prepupa, and emerges as an adult in the same season that the mother was also foraging. And depending on how far south you get, you can have many generations in a season. And so this is not so common up north, but the further south you get, especially in California and southern Arizona, you are going to see more generations of alfalfa leaf-cutting bees. And so keep in mind, especially for propagation, we want these overwintering bees because we want them for next year's pollination. If we have bees that are emerging in September at the very end of the alfalfa season, they're not going to have time to provision their offspring, and the offspring are not going to have the time needed to develop. So this causes a problem because we are losing that lineage or that offspring for the following year. So there's several theories on why the second generation happens. Climate is proposed as one. In the hotter generations, that that hotter temperature, the warmer temperature is signaling to those bees that they do have enough time to make another round of offspring. Photo period has some evidence that the longer days are a cue that there's enough time to forage and produce more bees and more offspring. There's some evidence that there is maternal effects where the mom and the foraging conditions that the mom is experiencing is being placed in the provisions, and that's determining whether or not they diapause. And then finally, the diet or the amount of food. So the quality and quantity of food has been hypothesized to be an indicator of whether these bees diapause or not. Now, the crazy thing is that within the same nest, you can have diaposers and non-diaposers, which leads to a couple of series of questions and kind of backs us up from these hypotheses to really understand what's going on at the individual level, at the nest level, rather than like a general species level. And so we were interested in two questions. What's the likelihood that siblings adjacent to each other would share the same diapause outcome? So you would expect that if the mom was choosing this, or if the conditions were such that they were responding to those cues, that any bee that was laid at the same time by the same mom would have the same diapause outcome. We know that is not true anecdotally, but we wanted to test this more rigorously and understand what is the actual likelihood that they have the same diapause outcome, diapause fate. And then, does the developmental rate change if the siblings have different diapause fate? So how likely are they to mirror each other, and at what time point does this differ? Okay, so to do this, we conducted this field work in the summer of 2023 in conjunction with normal flowering time of alfalfa, and we did this on a commercial alfalfa seed orchard in southern Idaho and northern Utah and in the US. And so on the top of this, I hope you guys can see this, we started in late June, went through August, and we had about 10 weeks of collection periods. And so we had two sites, one in Trenton, Utah, and one in Lab City, Idaho. We sampled 3 shelters at each of the sites. And at each of these shelters, we sampled from 3 boards, and we sampled the front and back of these boards to just get as much variety as possible. And then we have these 3 rows of straws. And we use the straws just so that we could easily pull out the offspring when we needed to collect them. And so when we got the bees, we brought them all back to the lab, and we were able to x-ray each of the nests. So these are the nests inside the straws. And what we were looking at were these sibling adjacent pairs, and we were really interested in understanding what happens with the females. So we only collected female pairs. And so to do this, we collected cell A and cell B, or the first laid pairs. And we tracked those bees through the development of their lifespan. And so if at any point these bees were parasitized or died, or we weren't able to track both pairs for the whole duration of their life cycle, they were thrown out of the study altogether. Sometimes we had these like ghost cells where there was nothing in them and it's clearly just the leaf pieces. In that case, we just started this as A. But like here, this was clearly a parasitized B, and so we didn't use this B and we didn't use this B. And because we knew this was A, we just like disregarded this whole cell altogether. And we continued to do this every day, or every 3 days for the duration of the study, we would x-ray them, we would see what life stage they were at, we would see how far they were developing, so we tracked their developmental time, as well as their overall diapause fate. So whether by early October, we were able to say, you are going to diapause, or you have already became an adult, for the non-diaposers. Okay, so what did we find? What's the likelihood that nest adjacent siblings share the same diapause outcome? So to do this, we just generally wanted to know how many bees diapaused or didn't diapause, and so you can see throughout the season. What we're seeing is that non-diapausers are getting less throughout the season, while diapausers are getting greater. This kind of corresponded with temperature, but it definitely corresponded with day length. So June 21st, the summer solstice, roughly is when this switched. As days got shorter, you got more diapausers. As temperatures got warmer, you also got non-diapausers, which is kind of counterintuitive to what we would have expected. This is consistent with what other literature has suggested about this pattern of diapausers and non-diapausers. Okay, so how likely are siblings adjacent to each other having the same diapause fate? And so early on in the season, early on in like shortly after the summer solstice, we're seeing that there is large-ish variability. So 76% had the same match fate, but that means 24% did not. Whereas towards the end of the season, you're seeing 100% match. So they are, towards the end of the season, you are seeing that they're all having the same diapause fate, and this kind of shows this little linear trend until it levels off. And overall, if you averaged everything together, you had about an 86% likelihood that you were getting the same diapausing fate as your siblings, which is pretty cool. So did the developmental rates change if siblings had different diapause fates? And the answer is yes. So here we see the duration from egg to pre-pupa, and in non-diapausers and diapausers. And what we can see is that from egg to pre-pupa, it took diapausers roughly 22 days, where non-diapausers, it was about 19 days. And so you are seeing a difference, and these are statistically different from each other, in how diapausers develop from non-diapausers. Okay, so what about siblings? If one sibling was a diapauser and the other sibling was not a diapauser, or if the siblings have the same diapausing fate, is sibling treatment more affected than whether your diapause fate? And what we found was, of those that did have these matching fates, so both either diapaused or non-diapaused, the difference in development of siblings was less than a day. So they were very, very closely aligned. However, if they had different diapausing fates, what you're seeing is that they did develop at this took longer to develop, or they had a greater difference in developmental time. They were not matching each other. And so really what this shows us is that whether or not the bee is diapausing is going to change its developmental time. We still need to it would be good to go back and break down, like, how long did it take to reach to the second instar? How long did it take to reach to that cocoon spinning? So we could break this down a little bit more, but what it's showing is that even in early stages, these bees are changing the way that they're acclimating to their environment, responding to these diapause pressures through their development so early on, we are seeing these differences. So there's a high likelihood that siblings have the same diapause fate, and this likelihood increases further into the season as more variable around the summer solstice, and nested adjacent siblings that have different diapause fate develop at different rates. So this is really cool because now, before this point, we didn't have a mechanism to really molecularly test these prior to diapause. Now we can use one as a validating pair, given the fact that most of the time you are going to see this relationship. You're going to see the same diapause outcome, so we can now begin to test this molecularly. There's this possibility that maybe it's not one thing that's affecting the outcome of diapause, but an interactive effect. And also, what we didn't do, what I would like to do in the future is look at the diet and provision size mass, because this has been hypothesized to matter more or just as much as maternal or environmental impacts. And so if the non-diaposers actually had less to eat, is that why they diapaused faster? Was it that larval stage where they were just consuming that triggered them to diapause? So that would be something that we're going to investigate further down the road. Okay, so shifting gears a little bit to talk about alfalfa leafcutting pest, because this is another major issue in the US and reasons why we can't propagate leafcutter bees here. So in the US, these are the 7 most common alfalfa leafcutting bee pests: Melittobia, Sapyga, Trichodes, Monodoctoromerus, Paramouse, which you guys also have, Trichodes, and bloated beetles. And so these, every location, everybody you talk to, you're going to be like, what pests do you have? But across North America, Western North America, these are generally the pests that you encounter. The interesting thing about these is that they attack different parts of the life cycle. Some are parasites, some are predators, some are opportunistic, some sit there and wait until the optimal time. And we know very little about most of these and their interactions. And one of the big things that we don't know is really seasonally, when do these bees occur? What is influencing their abundance, and how can we mitigate these pests? And so I took a step back and just wanted to know, all right, how abundant are they? When do they occur? And can we track their phenology somehow so that we can mitigate the impacts in the field? So to do this, I'm back into my 2 favorite sites in northern or southern Idaho and northern Utah. And once again, we are collecting these bees from a commercial alfalfa field through the growing season in June through July. And this was done 2 years, in 2022 and 2023. And so, very similar to what we did in the last site, we had the 2 sites, 3 boards, front and back of each board, and then we did something specific for this period, because I mentioned that all of these pests attack at different life stages. And so it was really important, had we sampled every week and just pulled straws, these all would have been really young bees, and we would not have maybe attracted the parasites that attack later on in life. And so we put straws in and sampled at different time periods throughout the season. So if you look at, this is just a picture of one of the boards, and you can see these rows of straws that we have, and then our timetable down here. And so we had 1 row of straws that was replaced every week. And so we would collect all the straws in that row, and every week pull all of them out, take those back into the lab, and replace them with new straws. And so these were our young bees. And then we had another row of straws that we collected every other week. And so this allowed those bees to progress further in the field, so further along in field conditions and be exposed to other parasites or other pests. And then finally, we had a third straw that we collected every 3 weeks. And this was really to get at those later life stage pests, because we really wanted to get a full picture idea of who was parasitizing these bees and when, or who was predating these bees and when they were doing it. Okay, so we have our collection periods where we have every week, every other week, every third week. And like what we did in the previous study, we took all of these bees back, we put them all on our sticky boards, and x-rayed every single bee every 3 weeks, every 3 days throughout the duration of the growing season, and actually through October, to understand what the outcome of these bees are going to be. And through our years of experience, we've actually developed a pamphlet, an X-ray book pamphlet, where we can tell exactly what is going on at either of these times in the X-rays, and be able to tell what parasite is infecting based solely off of the X-rays. And so this was really helpful because we could just X-ray these bees instead of do any destructive sampling, and we knew when we collected them in the field. Sorry, I forgot to mention this. All of the bees that were then collected, all the nests that were then collected from the field, were wrapped in organza bags individually, and so that there would be no pests that could get in or out. And because we X-ray these so frequently, we were able to detect, when they went in and when they were detectable on an X-ray. And so we're working on putting all of that together to get a more robust picture of phenology of these pests and then detectability on an X-ray. Okay, so what parasites, what pests did we see? Surprisingly, Monodontomeramus and Strastocus were not detected at all in the 2 years. However, Terramouse and Melittobia were detected at a very low level, and our shining stars in this case were Melittobia, Sapyga, and Trichodes. All right, so first we looked at seasonal changes. Just right up front, are there any seasonal changes of these 3 pests? And what we found was there was no effect with Melittobia in season. Sapyga had this bimodal effect where it was greatest in the intermediate part of the season, and Trichodes increased as we got further out into the season. And this actually was true for both locations. However, I will say that both sites in southern Idaho and northern Utah, despite them being in different states, were less than 30 miles from each other. So still generally close and in the same proximity to each other. And then we wanted to understand, all right, are there other environmental changes or weather indicators that could help us predict how these bees are going to respond, how these pests are going to respond. So we looked at mean, min, and max temperature, and mean, min, and max relative humidity. So I'm only going to show the results from data that was significant. So here we found, here we have the percent Melittobia and mean temperature. Sorry, I did this for another grower group and it's still in Fahrenheit. But what we found was that mean temperatures decreased Melittobia. So Melittobia was the most common in the cooler parts of the season. And if you remember, there was no seasonal change, but in cooler moments, we had an increased collection of Melittobia. For Sapyga, maximum temperature was the most important. And what we see is an increase in the number of Sapyga with very warm temperatures, and like these were hot temperatures. Even for our area, they were very warm. And again, so minimum temperature and mean temperature were not as important as maximum temperature was. So when it was those maximum temperatures were really high, you saw these increase in Sapyga. And finally, the minimum relative humidity. So the Sapyga seems to have these extremes going on where the hotter and drier it is, the more likely you are to see Sapyga outbreaks. And then finally, with Trichodes, minimum relative humidity was also important. And this was, the wetter it was, the more Trichodes there were. Okay, so just to kind of summarize this, Melittobia increased in cool periods, Trichodes increased in wet periods, and Sapyga increased in hot and dry periods of the season. Okay, so this is not like a smoking gun or anything because this was done relatively in a small geographic location, and we really don't know if this is going to hold true across the board. But from this, I did develop a growing degree day model for each of basically the phenology of the pests. And now the plan is to try to figure out ways to mitigate pest pressure prior to the emergence of, say, Sapyga, which is this lightest peaches color. You can see that there are actually two humps. And this does correspond with our first emergence and then the second generation emergence. So if we could figure out ways to like mitigate pest pressure here and here, that would be ideal. So I am going down that rabbit hole and trying to figure out ways to work on this. But what I really need is to expand the efforts. So most of this research has been done in a relatively small area, which allows for relatively small results and impacts. So what I really hope I can do this summer is expand our efforts. And so these pink dots are places that I'm already going to do this in the Western US. I would really love to expand this to this project to be a Western North America project and get sites in Canada. So if you are interested in helping me expand these efforts and you live anywhere in here or here, anywhere up here, please reach out to me. This can be as simple as I send you some leaf cutter bee blocks, you put them out for me, you send them back to me, and I will pay for shipping and everything like that, and deal with customs. But I am really looking to understand this pests, parasites, pollen ball, and second generation across the latitudinal gradient that is North America, and get some really concrete evidence on what's going on and how we can better understand and manage and mitigate impacts for our leaf cutting bees. So with that, I would like to thank all of my amazing team the last several years that has helped me accomplish this research. And if there's time, I would be happy to take any questions.
Does anyone have questions for Lindsie?
I have a comment. One comment.
I mean, okay. If I can figure out how to. Oh dear, what's town?
Spacebar?
I'm just, Lindsie, are you able to hear us?
Yes.
Oh,
Yeah.
okay. I just was curious if you're aware that we don't want any pests brought into the bees or anything. That's all. Like blocks couldn't come into us. No, nothing used is allowed into the bees. Yeah. Okay.
No bees.
Did you hear that, Lindsie?
No bees, no.
Sorry, I couldn't really hear that.
Okay, so it was just that they were wanting to let you know that so the piece is actually like free of a lot of the pests and stuff like that, and so we don't usually import, like, because we don't want new pests, like the pests that we don't have.
Chalk fruit.
Right, right,
Chalk
yes,
fruit is
that,
the biggest one.
yes,
So no, you
that makes 100% sense, yeah.
Yeah, so we don't bring anything used in.
But new materials could be used for sampling.
Yes, new materials can be used.
Oh, yes, yeah, of course, they would all be new, they would all be fresh, no issues there, yeah.
If we have time, Lindsie, I had one quick question about the sibling pairs.
Yeah,
And with the 25% where their diapause fate didn't match, do you have any sense of why that is? Like, I was wondering, is that as simple as an overnight pause in their cell creation work? Like, what do you think is causing them to be non-matchers when they obviously have a tendency to match?
yeah, so we have to look more into this, but I do think that the pollen provision size may be a key indicator in determining, okay, the mom is choosing to say, okay, I'm gonna give you less food, and then you will not diapause. And this could be like done in like variable weather scenarios, weather conditions where the mom feels like she may need to bet hedge more for her offspring to survive. And so we only did this 1 year. It would be really good to revisit this and do it again. It was like, it went from hot to cold very frequently. And so we may actually be seeing, there may be an increase, especially that year, where they don't match because the weather was unpredictable.
And I think Fred had a question.
Yeah, my question is coming from a neophyte. What is the difference in seed production between no bees, alfalfa leafcutter bees, and regular bees? Just, like, can the regular bees do anything?
Yeah. So just the question is that what is the difference in seed production between no bees, leafcutter bees, and regular honeybees or any other bee species?
Yeah, so this was done, there was a paper done like in the 70s about this, and I don't remember the numbers like crystal clear off the top of my head, but the no bees, there was basically no seed production. It was very, very small. Honeybees had like a slight increase over this, and then the leafcutter bees were like 6 times more effective than honeybees.
Yeah. From what I've been told, honeybees are lazy. They're
Yeah.
lazy pollinators.
They don't like to trip the flowers. They don't like to do the work. They just like to steal the nectar.
Yeah. Awesome. Are there any other questions for Lindsie? Perfect. Thank you, Lindsie.
So thank you guys so much.
Have a great day.